Hello everyone, thank you so much for joining us for this presentation on the composition and function of carbohydrates in the cell. This presentation is going to be presented by myself, Aubrey Mullen, and my partner, Richard Hedgepeth, and we are going to explain to you the background information for carbohydrates in the cell and how we plan to connect this information to students in the classroom. So without further ado, let's get to it. So what are carbohydrates? Carbohydrates are actually one of the most abundant substances that can be found in nature and are quite essential for all living organisms and their cells. They're known as organic macromolecules that are made up of only three key elements, which include carbon, hydrogen, and oxygen. There are also different types of carbohydrates that I'm sure most of you have heard of, and that would be sugars, starches, and fiber. As you see on the slide here, sugars are simple. They are the smallest unit of a carbohydrate. And then as the structures become more complex for these molecules, that's when you start to form starches and fibers. And then let me grab my pointer, you can see here that I've also listed some examples of each type of carbohydrate. So now that we know what a carbohydrate is, what is its composition really? And to break it down, it's primarily just the combination of carbon and water smushed together. So basically, carbohydrate gets its name from its composition. Carbon would be the carbo, as you can see right here, and water is a hydrate because it's hydrating the molecule, and that's where we get carbohydrate. And here you can see is how it would be formulated in the empirical formula, or basically it's the smallest molecular formula that we have for any of these carbohydrates. The N over here is just standing for the number of repeated units, which means that this is our basic formula that can then be multiplied. So our ratio ends up being 1 to 1 for the order of carbon, hydrogen, and oxygen. Now, the major carbohydrate structural classifications, so there's types and then there's classifications. The classifications are actually based on the structure of the molecules, and that's where we get our monosaccharides or monomers, our disaccharides, our oligosaccharides, and our polysaccharides. So since those are our types of structural classifications for carbohydrates, we're going to start from the simplest and work our way up. So as you can see here, monosaccharides are the simplest form of carbohydrates, and they actually have a common name known as simple sugars because they're only made up of one sugar molecule. Now, one sugar molecule can have up to between 3 and 7 carbons in their carbon backbones. The potential structure that this can create with these certain amount of carbons will determine whether it has a linear chain or a ring-shaped or what's known as a cyclic molecular structure. And these particular structures for monosaccharides are actually classified by the location of its carbonyl group, which is this C double bonded to the O, and then also the number of carbons in the carbon backbone. So as you can see here, for instance, this aldose, it has 3 carbons. Our glucose over here has 1, 2, 3, 4, 5, 6. So it can also be called a hexose, depending on what you're naming it for. Major functions for your monosaccharides, you can break that down between humans and animals versus plants. So monosaccharides in humans particularly are an essential energy source. It's actually the preferred energy source of the body. And what happens is when a human consumes sugars like glucose, cellular respiration actually breaks down the glucose, which releases energy, and then the energy is then used by other processes to make ATP, or energy for the cell. In plants, they actually produce glucose through a reaction with carbon dioxide and water. And this actually provides necessary energy that the plants can use. But not only that, any animals or humans that consume plants themselves also gain energy because they gain the glucose that the plants create. And if these animals and humans were to consume an excess of glucose, they can actually store it in the body to be used later as catabolized starch. Next, we have disaccharides. A disaccharide is where two monosaccharide molecules are chemically bonded through a dehydration reaction. Basically, during this reaction, a water molecule is lost and a covalent bond is gained. This particular covalent bond is called a glycosidic bond. And you can see over here in our illustration, in the blue box is that glycidic bond. Here is monomer 1, and here is monomer 2. When they're bonded together, they create that disaccharide. Some common examples of disaccharides would include lactose, maltose, and sucrose. Lactose is a disaccharide consisting of the monomers glucose and galactose, and is naturally found in milk. Maltose, also known as malt sugar, is a disaccharide formed by that dehydration reaction between two glucose molecules. And then we have the most common disaccharide, which is sucrose, or table sugar, which is comprised of one glucose and one fructose monomer. And then, as you can see down here, we have our functions. In humans, these disaccharides form or provide a quick source of energy. In this case, digestive enzymes are able to break down disaccharides into their monosaccharide forms, and that can easily be converted into energy for the host. In plants, these disaccharides can also help them store energy, as well as transport nutrients from the leaves to the roots and vice versa. Next, we have oligosaccharides. The structure length of these saccharides span from 3 to 10 sugar molecules. Some common examples of these types of saccharides would be raffinose and stachyose, both of which can be found in beans and legumes and are nutritionally important for the body, but it's very difficult for digestive enzymes to break it down into simple sugars. Some of the various functions for these type of saccharides would include the modulation of physiochemical properties such as solubility. It also helps with the cell membrane function, including transport across the membrane, intracellular migration, secretion, and cell recognition. It also helps protect against proteolysis and helps affect and allow for the development of embryos. Finally, there are polysaccharides, also known as glycans, which are actually large polymers that encompass 10 plus sugar molecules linked together by those glycidic bonds. Some important properties to notate for this type of saccharide is that, unlike their predecessors, these aren't sweet, and they're usually not soluble. Some examples would include glycogen, starch, cellulose, and an abbreviation called NAG. So glycogen is actually what's called a branched polymer, which is shown over here. This is kind of what it looks like, and this is what it looks like on the molecular level. A branched polymer, in this case, it's used for primary energy storage for bacteria and animals. For starch, which is also a branched polymer, is used for a primary energy storage for plants. So glycogen is for bacteria and animals. Starch is energy storage for plants. Then we have cellulose, which is also fiber. This is that linear chain of glucose, which this is kind of an example of a linear chain, even though this is showing amylose. But that's just kind of the basic structure that we're talking about. And cellulose is actually the structural component that can be found in cell walls, especially in plants. And then finally, we have NAG. It's actually a structural support for bacterial cell walls as well, but it can also be used to form something called chitin, which can be found in insect exoskeletons, as well as other various functions. So to summarize, carbohydrates are essential for all living organisms. They're composed of carbon, hydrogen, and oxygen at a 1-to-1 ratio, and they can be composed of one or more sugar molecules. The major structures can include linear chains, cyclic rings, or branched polymers. Major functions, for whether it depends on animals humans or plants, can include the following: main energy source. It could be a main storage for energy. It can be used for structural support, or in most cases, it can be used in the cell membrane to help with various other functions such as protection of the cell recognition, and cell transport. And now I'll hand the presentation over to my partner Richard, and he'll tell you how we're going to connect this information about carbohydrates to students in the classroom.
In this portion of our presentation, we're going to discuss activities and labs that enhance learning and understanding of the composition and function of carbohydrates in the cell. 3 different activities we're going to look at are carbohydrate testing, detection of carbohydrates, and sucrose digestion and hydrolysis. The first activity we're going to look at is carbohydrate testing. In our class discussions recently, we've talked about energy and its relationship to biochemistry. This exercise demonstrates and discusses the importance of carbohydrates in bioethanol or biofuel. In this exercise, students are going to learn how to ID carbohydrates based on their functional group property and structure. The goals of this lab are to utilize testing to determine carbohydrates and sugar present, and determining how sugar, which is located in the plant cell walls, can be released to maximize the amount of sugar to be converted into biofuels. Now, this has a very special place right now for students as the world continues to look at alternative fuels to replace fossil fuels. The next exercise for the lab we're going to look at is the detection of carbohydrates. This lab will be looking at detecting glucose using the Fehling reaction and fructose using the Sylvanov reaction. This exercise is designed to teach students how to detect the presence of carbohydrates using testing reagents. Quite frequently in the chemistry lab, reagents are used to determine the presence or absence of certain chemicals in solution. In this case, our students are going to be utilizing the Fehling reaction to detect glucose and the Sylvanov reaction to detect fructose. In this activity, students will look at the effects of enzymes on carbohydrate digestion. In this lab, students will take 2 samples of a 2% sucrose solution in a test tube and add invertase to 1 sample. Students will then mix these samples and boil them in a water bath over a period of time, and then measure the amount of remaining sucrose in each sample. In this exercise, students will learn how enzymes affect digestion of carbohydrates, and the goals are to determine the effect an enzyme has on carbohydrate digestion by measuring the amount of sucrose consumed in the presence of an enzyme versus a sample with the absence of enzymes. In this final section, we're going to take a look at assessments, assessing understanding of the composition and function of carbohydrates in the cell. Now below we see different types of assessments that look at carbohydrates' role in relation to plant and animal cells. The first one on our list is our typical go-to as teachers, and that is quizzes and tests. But that's not the only way to assess a student's understanding. Another activity could be to create carbohydrate models. Give a student a list of different carbohydrates and have them utilize beads and pipe cleaners to build carbohydrates, not just monosaccharides or disaccharides, but in polysaccharides as well. Another example could be written assignments. We all have had formal papers on topics, and of course this is a legitimate way to utilize, or rather assess, the student's understanding of the topic. Another activity might be send the student home, have him do personal carb counting during a week, come back in and calculate the amount of carbohydrates and sugars that a person ingests, or rather that student ingests, over a week's time. This could help the student understand the role of carbohydrates and how important it is to monitor the intake of sugar and carbohydrates in a student's diet. The next one, of course, could be group projects, as we have here today. Get students together working on projects that, again, illustrate the composition and function of cells, or rather carbohydrates, in the cell. And finally, presentations. Just like we're doing today as well, presentations that consist of either PowerPoints, live presentations in front of the class, or other methods. Presentations represent a great way to assess a student's strength of understanding on a topic. That wraps up our presentation on the function and composition of carbohydrates in the cell. Thank you.
And there you have it. Thank you guys so much for taking the time to watch or during my presentation on the composition and function of carbohydrates in the cell and how we plan on connecting this information to students in the classroom. We hope you enjoyed it and could take something away. And until next time, we hope you all have a beautiful and safe day.